common cloning vector, and that's what you see on the left-hand side here, where they cut out the genomic DNA from the species you're studying, and the little bits are put inside plasmids, and those are transformed inside the bacteria. This other cloning vector is bacteriophage, which, remember, bacteriophage is just a type of virus that infects bacteria. And remember, viruses have their own chromosome, and so what they can do is ligate the gene of interest into this bacteriophage's chromosome, which you see going on there, and then they put these recombinant viral chromosomes inside capsids and then allow them to infect bacteria. And yeah, that kills off the bacteria, but that allows the viruses to be reproduced. And so again, you end up making millions of copies of each of these recombinant bacteriophage. And again, that is what cloning the gene is, making millions of copies of it. There's kind of a neat picture I found somewhere of a bacteriophage. Almost looks like a hypodermic needle injecting its DNA, which is the red stuff there, inside a bacteria. Okay, so there was this other type of library called a cDNA library, which is different from a genomic library. And here's what those are all about. This represents a chromosome, maybe a jellyfish chromosome or a human chromosome. And of course, each chromosome contains hundreds, if not thousands, of genes. There's gene number 1 on the chromosome, gene number 2, gene number 3, and there would be gene number 4, and 5, and 6, and 7, and 100, and 1,000, all down the chromosome. Most cells in your body are not using all those genes. Someone estimated that only about 20% of the genes are being used in any particular cell in your body. You know, you may wonder why. Well, some of these proteins might be for eye-specific, genes might be for eye-specific proteins, like the proteins that make up the lens of your eye. You know, a cell in your fingertip doesn't need to express those eyeball proteins. That wouldn't make any sense. So it's just not going to be expressing that gene. And if this gene might be for an enzyme that digests your food in the stomach, you know, it wouldn't make any sense for, I don't know, a cell in your spleen to be expressing that particular gene. So the fact is that only about 1 out of every 5 genes is being expressed in any particular cell. And so sometimes people might be interested in just the genes that a particular cell type is expressing. You might say, I just want to know about the genes that are expressed in stomach cells, and I don't care about any genes that are not being expressed in stomach cells. And so if that's the type of thing you're interested in, a cDNA library would be the type of thing you would do. Now, what's the difference between genes that are expressed and not expressed? Well, the genes that are being expressed, messenger RNAs are being made from those genes and not from the genes that are not being expressed. And so to begin to make a cDNA library, you isolate the messenger RNAs from whatever tissue type, like stomach tissue, that you're interested in. So initially, you've got a bunch of messenger RNA molecules. But there's this enzyme called reverse transcriptase, which can take RNA molecules, and from those RNA molecules make a DNA molecule out of them, make a complementary DNA called a cDNA out of them. You might remember this reverse transcriptase enzyme actually was isolated from a virus, a type of virus called a retrovirus. But it's available to molecular biologists now. So the molecular biologists take the RNA they've isolated and add the reverse transcriptase enzyme to it. And you can think of it as essentially converting the RNA into DNA. It doesn't actually convert it, but it uses the RNA as a template to make a complementary DNA. So now you get these DNA pieces based on your RNA pieces. And these are now called complementary DNAs or cDNAs. And you can clone these into a cloning vector like a plasmid. And again, once they're inside the plasmid, you put them, use the plasmids to transform bacteria, and then as the bacteria grow, the bacteria act as copy machines for the pieces of DNA that you've put inside there. So, what might a person use a genomic library versus a cDNA library for? Well, let me ask you this. Remember, the cDNAs really come from the messenger RNAs. So do they contain promoter sequences? The messenger RNAs contain promoter sequences? No, because remember that the promoter sequences are not transcribed. The RNA polymerase binds the promoter sequences, but the transcription doesn't begin until about 30 nucleotides downstream of the promoter. And so a cDNA library would not give you any promoter sequences. And also, since cDNAs come from messenger RNAs, they don't have any they don't have any introns in them, because the introns get spliced out before you get to the messenger RNA stage. And so you're getting what are essentially the exon sequences, the protein encoding sequences in a cDNA library, but you don't know anything about the promoter, what I call the regulatory sequences here, or the introns. And so you really wouldn't know anything about alternative splicing or the promoter region of the gene. But you do get the protein sequence out of it. And if that's all you're interested in, that's a fine thing. But if you do want to know about the splicing and the promoter activity of the gene, then you have to get the genomic library, because the genes do contain the promoters and the introns and the exons and all the regulatory stuff. Alrighty. Okay, well, let me think. I'll shut down the computer here and now kind of redirect us, so to speak, more along the lines of what you're actually going to be doing in today's experiment. Give me just a minute to shut things down here.